Our brain is just like the universe. Not fully understood or discovered. We only know the basics. In fact, scientists only understand how 10% of it works. There is yet to be a theory established on how the brain actually functions. The brain is made up of two main types of cells: neuron cells, which help send and receive nerve signals, and glial cells, which provide support. One of the main types of glial cells are astrocytes, which specifically help regulate neurotransmissions by intimately surrounding synapses. Now, this is where it gets a little tricky. Neuroscientists have long suggested that astrocytes may have an active role in the synaptic transmission. However, this theory was thrown in the garbage bin because there was no evidence to support it. However, Andrew Volterra and team set out to find the main machinery responsible for the rapid release of glutamate in the brain, the main neurotransmitter released by neurons. For their study, they used the brains from 8 different mice. They put them through a process called single-cell RNA sequencing. This is where they collect a tiny sample of brain tissue from each of the mice and separate the tissue into individual cells. Inside each cell, they extract the RNA from the DNA, which is responsible for carrying instructions to make proteins. The RNA is then converted to complementary DNA, and each of the strands get a unique color assigned for recognition. Then a sequencing machine reads the genetic information from each cDNA molecule. Using a powerful computer program, they analyze and look at which genes are active in each cell to see how they function. One of the cells was an astrocyte cell, and they noticed a protein called vGlut1 was responsible for glutamate release specifically in that cell. This is how a new type of cell was discovered in the hippocampus part of the brain, known as glutamatergic astrocytes. The scientists tested one more thing. Without vGlut1, memory and epilepsy decrease. With it, they increase, proving that glutamatergic astrocytes actively release glutamate. Now, why is this important? Finding glutamatergic astrocytes will lead to new discoveries in research, neurological disorders like Parkinson's and Alzheimer's, as well as other therapeutic benefits, leading to new pathways in neuroscience.